Our cells are essentially a compilation of metabolic reactions, but left on their own, reactions are actually probably not beneficial to the body. And the reason is that left on their own, some reactions will go when they're not needed. Maybe we don't actually need that product, or cell's not ready for that product. We already have too much of that product. And some reactions won't go when you need them to go because they require an energy investment or because they are non-spontaneous. And that's where enzymes come in. So enzymes work essentially to manage the chemical reactions in a cell. And in general, we say that enzymes speed up metabolic reactions. They can do both. They can also block reactions from happening, but we're kind of going to assume that the main job of enzymes is to speed up chemical reactions, metabolic reactions in the cell. So an enzyme is a macromolecule. It is a protein that acts as a biological catalyst. Now, there's two definitions right there. One is used to define the other. A biological catalyst is an agent that speeds up a reaction without being consumed. In other words, enzymes regulate the speed of metabolic reactions. And we'll see in a minute that they do this by lowering energy barriers. In other words, they work to allow the cell enough energy to produce those non-spontaneous reactions. They will also manage spontaneous reactions because sometimes a reaction is spontaneous and it is going to put out energy, but it might move too slowly for the cell left on its own. So I have an example here. This is an example of the hydrolysis of sucrose. And if you remember from chapter 3, hydrolysis is how we break down our biological molecules. It's called hydrolysis because in the process we split water. And it is essentially the opposite of a dehydration reaction. So in this process of breaking down sucrose, the enzyme sucrase is actually going to bind to both of these products and essentially bring them together so that they can react, so that we can cleave this bond in the sucrose and produce glucose and fructose. Now we want to do this because glucose and fructose are what the cell uses to produce ATP. This enzyme, sucrase, is key in making that happen. Anything that ends in an ASE is an enzyme. So if you see that word, you automatically get a clue about to what its job is in the cell. Sometimes they make sense, like the enzyme that breaks down sucrose is sucrase. Sometimes the names don't line up. It is what it is. So all chemical reactions require an activation energy. Now, this is whether or not it is non-spontaneous or spontaneous. A spontaneous reaction is going to put energy out, but it may require a little energy investment to get the reaction started. So activation energy is simply that energy investment. It is the energy required to start a reaction, even if it's going to release energy. So I like to illustrate it this way. Let's say you're at the top of a hill on a playground and you want to roll a ball down to your friend at the bottom. Now, you might be at the flat part of the hill at the top and you set that ball down. It's not going anywhere. Eventually, it's going to roll down the hill, but you have to put in a little bit of energy. You have to give it a little push before it will roll down that hill on its own. That is your activation energy, that little bit of energy push to make the reaction happen. So enzymes work to enzymes manipulate bonds to lower activation energy. So that's where our analogy breaks down just a little bit. Enzymes don't actually add energy, they just change the system so less energy is required to make that reaction go. And that's really important. We're not putting energy into that reaction, but we are putting in an investment, which is the enzyme's function. So that activation energy can come from two different sources. It can come from heat from the surroundings, or it can come from enzymes lowering that activation energy. Now, heat is bad for cells. There's going to be some ambient heat in a cell that can provide that thermal energy for activation. But cells cannot continuously become hotter and hotter in order to supply all of the activation energy for all of the reactions that are taking place. Heat is not good for cells because it can interfere with the structure of the plasma membrane. That plasma membrane is fluid and it's a lipid. And when you heat lipids, they become more fluid. Think about butter in a pan. When you heat it, it melts. Cell membranes are the same way. If you heat them too high, they can become too fluid. So we can't continue to add heat from the environment. That's where enzymes come in. Enzymes can lower activation energy so that we don't need as much heat in the cell. So over here on the side, we have got our reactants. We've got our energy level of our reactants, which is higher than our products, which tells us that this is actually a spontaneous reaction. So that reaction is spontaneous, which means it's going to go and it's going to release energy. Now, we also have this little hump in the middle. This hump in the middle represents our activation energy, the amount of energy that is required to get that reaction to go. So maybe it's supplied by heat in some circumstances. Maybe it needs to be regulated by enzymes. So enzymes work to make the bonds between the reactants unstable. We'll talk about what they actually do in a minute. And when they make them unstable, they are more likely to react. And so enzymes work by binding the reactants, making them unstable, in which case they actually need less activation energy so that they will react to produce our products. So enzymes work so that in a way that they change reactants so that they can absorb enough energy for the reaction to proceed. So basically, they lower the amount of energy investment so that the heat provided by the cell is enough energy for that reaction to take place. Now, one important thing to remember here is that enzymes do not add energy. They change the stability of the reactants so that the reaction is more likely to occur. Also, enzymes change the rate of the reaction, not the energy produced. So the reactants have an amount of energy, and even though some energy might be put in for activation, they're still going to release the same net amount of energy. So enzymes aren't changing how much energy is coming out at the end, only how fast that reaction actually goes. So in a non-spontaneous reaction, enzymes are going to help with energy, not add it. They're going to make the reaction more unstable so that less energy is required. In a spontaneous reaction, they're basically going to jumpstart the reaction. by lowering the amount of energy that is required. So if we look at this graph on our right here, we can see the energy of the reactants. The black line is the reaction without an enzyme. So without an enzyme, it requires that much energy from the level in the reactants to the activation energy. Now, an enzyme is not adding energy. It's just lowering the amount of energy that's required to make it go. So once that enzyme lowers that energy level, the ambient energy in the cell from heat can cause that reaction to go. Now, the net release is still the same. We still have, in this case, a spontaneous reaction because we have energy release. Because the net release is negative. So before we can start talking about how enzymes actually work, we need to define a few things. First off is the substrate. Now the substrate is the reaction that the enzyme acts on. Reactant. So over here on the right, we have our enzyme, that's the big blue molecule, and then we have our substrate. It's that little red molecule there. In our example from earlier with sucrose, sucrose would be our substrate. That is the reactant, that is the molecule that our enzyme, sucrase, is going to act on. So we could say that this is sucrase. Now the active site is the region where the substrate binds. So here it is this little notch in the center, and substrates and enzymes are very, very specific. There is usually one enzyme and one enzyme only for each type of chemical reaction. So this sucrase molecule will only recognize sucrose. It won't recognize something similar. It will only recognize sucrose. It won't recognize glucose, which is only part of it. It has to be the whole molecule. So enzymes are extremely specific. Once that substrate binds the active site, we form what we call the enzyme substrate complex. So it's just the enzyme and the substrate. When that happens, we get something called induced fit. With induced fit, the active site changes shape and tightens around the substrate. And it is in that active site where that induced fit has closed around the molecule that our reaction is actually going to take place. Now, the way that that reaction takes place depends on the reaction itself. So these ways that enzymes lower the activation energy is going to change. There's not one way, but there are several different ways that they might happen. So enzymes can lower activation energy in several ways. One, they can simply bring the reactants close together. And then when they're near each other, they're more likely to react. It's basically like friends at a party. When you come together, you interact. Enzymes can also do something to make the reactants unstable. So they might bend or stretch the molecules so that they're not stable anymore. And then once they reach a certain level of instability, they're going to react because they're being stretched in a certain way. An enzyme might also simply provide an isolated microenvironment for a reaction to happen. Maybe the reaction actually needs to happen in an acidic environment and your cell is kind of basic, or vice versa. And then enzymes may also occasionally actually covalently bond to the product, and that might be the source of the instability. Each reaction is going to have its own way that it goes. So a single reaction might happen because the reactants are brought together. A different reaction might happen because the enzyme is working to stretch the molecules' bonds and make them unstable. So, how do enzymes actually work? Well, first, we might have an enzyme, and we might have two substrates. So let's say we want to make a reaction where the round substrate combines with that blue substrate. So those guys are going to enter the active site. And that active site is going to bind to those molecules. I'm going to fill them in so we can see them. Now, that enzyme has brought those reactants together. So let's say that inside they became unstable. So maybe this enzyme has stretched some bonds and now they want to react. So we have our enzyme now, and we have our reactants that are unstable. And when they are unstable, they react to form our product. Now, the enzyme can go on to perform another reaction. That product is going to be released, and then that enzyme can go and bind some more reactants. Because remember, an enzyme is a catalyst, and a catalyst is something that participates in a reaction but is not consumed. So this enzyme can go on, bind more products to continue to produce more reactants. Now, this product is more stable than those reactants that were made unstable inside that enzyme. So on the previous page, I said there were 4 ways that an enzyme can work. And I said that sometimes it creates a little microenvironment. So this might be a sheltered area for that reaction to occur. Or it might just be the area where these guys are in close enough proximity just to react. So here's our book example of this. I like to draw things because I hope that you draw them together with me. But your substrates are going to bind at your active site to produce that enzyme substrate complex. It's going to bring those reactants together, cause the reaction to happen, release those products, and then that active site can now bind new substrates. So enzymes are proteins, and there are a lot of things that can affect the function of a protein. Oops, I'm sorry, I got ahead of myself. Oh no, I didn't. Okay, so the substrate is going to hold those reactants in place, usually by a weak chemical reaction. Enzymes are going to work to push forward that reaction. An enzyme can also sometimes do the backwards reaction. It depends on the enzyme. So the same enzyme that is breaking something down might also be building the same thing in the other direction. But as I was saying, enzymes are proteins, and proteins are very strongly affected by the environment that they're in. And this has to do with the tertiary structure. Actually, all of the structures, the primary, secondary, and tertiary protein structures, because those structures are made up of a variety of different types of bonds. Some of those bonds are hydrogen bonds, which are interactions, not covalent bonds. Others are attractions between positive and negative regions, and all of those things can be interrupted. And in chapter 3, I believe, I hope, I very briefly talked about denaturing. Denaturing is when a protein structure is interrupted. So that is caused by breaking down any of these tertiary, secondary structures. If a protein is denatured too far, it won't go back into the right shape and it becomes completely dysfunctional. Sometimes it's just mild denaturing and can actually come back and be functional again. It depends on the situation. So really, anything that can affect the environment that a protein is in can affect its function, because its function is specifically related to its actual structure. So if you interrupt the structure, you can interrupt the function. So let's look at some of the ways that we can affect enzyme activity. The first is concentration. So if you just increase the amount of enzyme or increase the amount of substrate, you're going to initially increase the reaction rate. If you increase them both at the same rate, it doesn't affect the rate of the reaction. But if you add more enzyme, there's more enzyme initially to bind the substrates and make the reaction go. If you increase the substrates, there's more chance that those substrates are going to find enzymes. You can come to a point, though, where the reaction is limited. And at that point, your reaction rate is just going to depend on how fast the enzyme can go, how fast the enzyme can actually find, bind, and cause the reaction. Because at some point, there is going to be a finite number of molecules. It's like having a finite number of people to do a job. At some point, everybody is max performing, and you can't do your job any faster. Temperature can also have a big effect on enzyme activity, and that is because temperature can interfere with protein structure. So usually proteins work best under optimal conditions. When we're talking about cells, we're usually talking about body temp. So at normal body temperature, those enzymes are going to work best. Temperature can also simply increase the activation energy. So we might raise the temperature a little bit, and that just causes the molecules to move more quickly. I'm sorry, I didn't mean to say increase the activation energy. Temperature can increase thermal movement. So aside from the enzyme, an increase in temperature can just cause the molecules to move around more quickly, and it will also provide some of that activation energy. So we wouldn't necessarily need an enzyme to lower activation energy if we can put heat into the reaction. And if those molecules are moving more quickly, they're more likely to bump into each other, to encounter each other, and therefore the reaction is more likely to take place. So temperature can initially speed up a reaction. You can do the same thing with lowering the temperature. You can decrease the reaction by just reducing molecular movement. When temps get too high, you can denature your protein. In other words, you can cause that protein, that enzyme, to come apart and become dysfunctional. So there's usually an optimal temperature range that's a balance between moving those molecules a little bit faster and getting that enzyme to work quickly. Because if you get too hot, you can denature. If you get too slow, you can essentially freeze everything. Too cold, you can essentially freeze everything from happening. pH is also really important in a reaction. Changing a pH in either direction neither one is optimal. So you can't increase a pH or decrease a pH necessarily to change enzyme function. pH affects enzymes in a certain range. If you get outside of that range, whether it is up or down, you're going to decrease enzyme function. And that's because acidic and basic environments interrupt function. They interrupt the structure of the protein and therefore the function. So most enzymes that we are going to encounter in a cell are going to be most active between 6 and 8, a pH of 6 and 8. So roughly neutral. They are all going to have a specific pH at which they work best. Maybe it's closer to 6, maybe it's closer to 8. And when you get outside of that optimal range, they're going to be less functional in either direction. There are some specific cases where pH is a little different, like in our stomach, it's a very acidic environment. The enzymes that work there work optimally in an acidic environment. We also have cofactors. So cofactors are non-protein helpers that bind to the enzyme. So they might be required for enzyme function. They might just make the enzyme function a little bit better. But these are usually inorganic molecules. They can also be organic molecules. Some examples are vitamins and other essential nutrients. And they're usually required in really small amounts in our diet. So these are things that we're probably getting in our diet just by normal consumption of food, but that are super important as cofactors in a chemical reaction. We can also have something called an inhibitor. So an inhibitor is a molecule that blocks enzyme function. Now, this is usually on purpose, and it's usually a regulatory mechanism. So these are naturally occurring substances that their purpose is to regulate the reaction. And it's usually so that the reaction doesn't keep going when it's not supposed to. But you can also have things that negatively affect a particular reaction, like an inhibitor that's not really supposed to be there. There are two types of inhibitors: a competitive inhibitor and a non-competitive inhibitor. Now, both types of inhibitors, are going to bind to the enzyme and cause a shape change that makes it dysfunctional. So these inhibitors, bind the enzyme and change its shape. And when its shape is changed, the active site changes and it can't recognize the substrate anymore. Now, there are two different types. Competitive inhibitors will actually bind at the active site. So they compete with the substrate for that active site. And so we see that here in the center. You've got this competitive inhibitor that is bound to the active site, not perfectly, but is bound to the active site and blocks that substrate from coming in. We can also have a non-competitive, and a non-competitive inhibitor binds elsewhere. So somewhere that's not the active site, but when it binds there, it causes the structure of the enzyme to change so that that substrate doesn't fit. So they block enzyme function by binding to the enzyme and changing its physical structure. Finally, We can also affect enzyme activity by different types of basically feedback loops or binding of specific molecules in order to regulate. One example is called feedback inhibition. And in feedback inhibition, the end product actually binds to the enzyme to block function. So this is going to happen when products accumulate. So this metabolic pathway is for the production of isoleucine, which is an amino acid. And so you can see here we start with threonine that binds to the active site, and then it goes through a series of steps and enzymes to get to the final product, this isoleucine amino acid. Now this pathway is going to continue until there's enough buildup in the cell, which means there's enough isoleucine for the cell that that isoleucine binds to the active site and blocks it. So essentially, actually this one is binding at a non-competitive. I was thinking of a different example. Non-competitive site. So it is binding here once it accumulates. So the concentration of isoleucine in the cell gets too high, and it works basically a feedback loop to stop that enzyme actually producing more isoleucine. This site that the isoleucine binds to, this non-competitive site, is also called an allosteric site. So an allosteric site is specifically for that end product. It's working the same way as a non-competitive inhibitor by binding that original enzyme, causing it to shape change so that we no longer convert threonine into isoleucine.